from Alex Kulikov, my colleague here at Cold Spring Harbor, who will tell us about DeepNose.
Thank you very much. Thank you for perseverance and for staying around. It's a pleasure. I'm a local person here. My name is Alex Kulikov. I do computational neuroscience here. I'll talk today about olfaction. I have a video here. Alex Velichko gave a wonderful presentation, so this is just a very basic introduction. I should say that nothing in this video is up to scale. So the molecules are not really that big. So they fly into our nose and then they interact, they land into the olfactory epithelium where they interact with those neurons, which are called olfactory sensory neurons. And they're colored with the different colors, and that signifies that they express a special protein, which is called olfactory receptor, that interacts with the molecules, and then they get activated, and they carry spikes into special structures in the brain called glomeruli. Glomerulus, as I learned, is a sphere in Latin. So they tessellate the surface of olfactory bulb. In the mouse, there are about 2,000 glomeruli. In humans, there are about 6,000. Nobody knows why. But the important thing is that the neurons that express one receptor, they converge on a single glomerulus. So there's this wonderful segregation of channels, and then they convey this information about which molecules are present in the mixture to the principal neurons of olfactory bulb, and those neurons carry this information to the cortex, which this is appearing in a second. And then on the basis of that activation pattern, cortex forms a percept of the actual smell, which will emerge in a second. So this is basically how olfaction works. There's a flower, apparently, corresponding to those molecules. To summarize, there are sensory neurons that express those olfactory receptors, which are specialized proteins. We have about 350 of them. So there are about 350 colors. They converge on discrete structures called glomeruli, and there is neural networks that kick in, and they eventually produce the odor percept. So this is just in comparison of olfactory genomes, of how many receptors different species have. This would be like our vision. It has three colors. In our olfactory system, there are 350 intact genes, and there are those pseudogenes, which majority of our olfactory genome are dead or extinct genes. So basically, if I compare our vision to the olfaction, if vision has three colors, about 1 million pixels in the eye, the olfaction has 350 colors and about 4 pixels. And so on. And like mice have way more receptors than we do, so presumably they have way more sophisticated sense of smell. And so we're attempting to solve this, attempting to figure out how this thing works. Right? So this is one of the hypotheses of what olfaction does. So we have a bunch of molecules. When the olfactory system is exposed to those molecules, the olfactory receptors, those odorant receptors, which are the proteins and corresponding glomeruli, get activated. And this pattern of activation is transmitted into the so-called olfactory manifold, or odor space. So it will be just a point on this manifold, which has presumably some small number of dimensions, way fewer than the number of receptors, and it's an analog of RGB space in color vision. So that would be one potential way how this can work. I mean, this idea has been developed for a while. I'm just like flashing out. There is a long history of the studies of the so-called odor space, which is an analog of RGB space. We had a meeting in Hanover about 10 years ago. The first most important paper, as far as I'm concerned, was this paper by Susan Schiffman in '74, which was, as far as I know, was the first application of dimension reduction techniques to olfactory perceptual data. And then eventually this paper in Science, which Alex Velichko was talking about, it is definitely perhaps one of the ultimate achievements in this field. So, but this idea has been around for a while. The idea of olfactory manifold or odor space. So just to give you some idea about how data looks like, I will talk about this like fairly old publication that we published some years ago, but I think I'll dedicate a few minutes just to describe the data sets, the typical data set. So this is Andrew Dramniak's. It's called Atlas of Odor Character Profiles. Each page in the book is dedicated to individual chemicals, so the chemical is listed here. So olfactory data sets in humans are mostly semantic. So they cast smells into semantic space, so which comes with its limitations, but I mean this is the most we know about how smells smell. And then for each word, there is a number, and this number is percent use. So basically percentage of people in the army of observers. There's 100 people here contributing to this point. It's 16% of the people thought that this chemical smells cool, and so on. This data set requires enormous effort to collect. So it was published as a book. There are about 144 odors. Each of them is present, cast into 146-dimensional space. So this is one of the highest quality data sets that we have at this point. Right? So you can try to embed this data set in a low-dimensional space. So this is this is the same basically embedding. I'm showing it from two directions. And the individual points here, the crosses are the perceptions of odor, those numbers that I showed you. Yes, and the circles are those points on this projected on this manifold, which is in this case a two-dimensional manifold. We call it a potato chip. I colored here the manifold as red and blue, reinforce that metaphor. Interestingly, this two-dimensional manifold captures about 94% of variance in 3D and about half of variance in the entire data set. So which kind of motivated us to propose that perhaps olfaction is not so high-dimensional as it may appear. So the high dimensionality of our smell is probably an illusion, because as you move along this two-dimensional manifold, you will see different percepts changing, but in fact, the number of parameters that I needed to define that smell is smaller. So this is trying to expand the manifold to more dimensions, and so this is included variance as a function for the solid lines are for the testing data, for held out data. So this is a two-dimensional manifold. The potato chip that captures about 50% of variance. The black line is important. And then this is a three-dimensional potato chip, which we call potato. And then there are four-dimensional hyperspot, and so on. And as we increase the number of dimensions to 10, the variance saturates at about 80%. So that was very exciting to us, which suggested that we actually have almost exhausted the variance in the data set. So what is the significance of those dimensions of x and y? I can put principal component loadings high in one direction and high in the other direction, and try to just eyeball them and see what's going on. And if I read the ones which are on top and at the bottom, I will see immediately that they signify some form of pleasantness or valency of odors. I mean, this is very, very well known property of odor perception, that perhaps the most important variable for us is if I am to define it cognitively, it would be the valency. And the horizontal dimension is a bit more controversial. You could read those words, and I basically asked a bunch of people what they mean. So, there were some hypotheses like woody, resonance, burnt, musty. It's probably debility, maybe something associated with kitchen. But the most exciting and perhaps controversial hypothesis was that it's auditory pitch. So apparently some people can say that these words or these smells are associated with a lower auditory pitch than these ones. So chemical, gasoline, fruity citrus, sour, vinegar is probably higher pitch. So this is a remarkable example of synesthesia. Perhaps the person who discovered this order to auditory pitch synesthesia was a perfumer who worked in London, whose name was Septimus Pease. So this is a book by his son, Charles Pease, and he created the so-called gamma of odors, where he arranged smells on the musical notes, and then he composed perfumes by playing music. So that was a bit interesting, but he was a very famous perfumer. at his time, like about mid-19th century. And so he said that in this following gamut, I have endeavored to place the name of odors in its position corresponding to its effect upon the olfactory sense. And he says that he virtually can put any odor on the frequency scale. So this synesthesia is not so far-fetched. Interestingly enough, this horizontal dimension that I pointed out, it's very easy to relate to actual neural circuit because it relates. It looks like it's smells which are highly polar to oily smells that are less polar, hydrophobic versus hydrophilic. And there is amongst olfactory receptors, there are two classes. There are fish-like olfactory receptors, which are called class 1, and there are the ones which are exploded in terrestrial vertebrates, which are called class 2. And perhaps this dimension just measures the polarity or the difference between class 1 and class 2 receptor outputs. Just to finish this, I'll take you a little bit off topic. This perfumer, Septimus Peace, is kind of famous because he contributed to this. It turns out that you can actually smell the perfume, you can buy the perfume that he's made, or replica of it, which was lifted off the shipwreck of a ship which was called Mary Celestia, which drowned off the coast of Bermuda, and then somebody found it. So the reason why the perfume, it was made of natural compounds. It's very challenging to preserve. This ship drowned about 160 years ago. So they found a closed bottle in this shipwreck, and somebody reverse engineered the perfume. You can just buy it there. It's called Mary Celeste in this perfume store, Lily Bermuda. And this perfume was made by Septimus Peace. So we're trying to build machine learning models for this. Now, the problem that we're trying to solve is to cast the chemical structure into the semantic space. So the data sets that we're dealing with, they're mostly describing smells with the binary numbers, like whether a given word applies or not. So they're less high quality than the one which I described before. So these are the 4 data sets. They include thousands of smells. Each of them is described by potentially hundreds of descriptors. So this is like one point in this data set where you have this molecule with the given CID, which is described as sweet, musk, and floral. And this is what we have to predict. So like a standard way of dealing with this problem would be to build a GNN, or graph neural net. This is something that a conventional way how people deal with molecules in computational chemistry. So we took a little bit of different route. We took the neuro AI route, which means that we're trying actually to build a network that with architecture that mimics the olfactory system, because we probably will integrate some other sources of data into this model. So at least we're hoping that at some point we'll be able to do so. So just to remind you, the olfactory perception is relying on the responses of those olfactory receptor proteins, which take the molecules, they bind them according to their shape, like matching shape in the molecular pocket, and then they convey this information to the rest of the olfactory system. So the hypothesis that we pursue is that those olfactory receptor proteins can be viewed as convolutional filters that interact with molecules in 3D, and they extract, like similarly to convolutional networks that solve problem in the image recognition, they extract this information about the three-dimensional shape of molecules and then convey it to the rest of the system. So we therefore decided to build the convolutional neural net. The issue with convolutional neural net is not explicitly rotation invariant, and so we tweaked the architecture in such a way that it becomes approximately explicitly orientationally invariant or approximately equivariant. And it also mimics the interesting distinguishing feature of the olfactory perception is that at the same time, olfactory system faces many stimuli. So each molecule is an individual stimulus, and it has to be recognized. And so there are multiple copies of the same molecule which are present. and those copies can approach olfactory receptor at different orientations. So this is just to introduce our network, which is called DeepNose. We marketed that word because we were the first. So the inputs into the network will be 5-dimensional tensors. And the first 3 dimensions are kind of trivial. It's just X, Y, and Z of the molecule. So this molecule will produce those like clouds of activation cast onto the rasters. 1 dimension would be the feature dimension, which is in this case just element. What element is present at this point? So carbon, hydrogen, chlorine, and so on. And another dimension will be the orientation. So this will be like right now in training we use 640 orientation in random directions, which are just sampling the orientational space. So in inference we can use more orientations, but so essentially the same molecule is sent through the network in 640 copies. So and as you rotate the molecule, the representation of the molecule, like for example, these are 2 representations of the same molecule, which are flipped by 180 degrees, but there are more. It's more continuous. The space is sampled in a more continuous manner. As if you rotate the molecule, the orientation, the presentation of molecule will just shift in that space. So the network is approximately equivariant. So we call it equivariant CNN. So this is the overall structure of the network. So the molecule is this 5-dimensional tensor that is sent into the convolutional layer with multiple orientations. And then at some point, we sum up, we pool over all orientations. So representations of the same molecule will be combined, so averaged over all orientations and spatial positions. And then we have something which resembles this olfactory receptor channel, which is the hidden representation, which is supposed to mimic what olfactory system does to the odorants. And then there is a few layers of MLP, and then eventually we have the semantic output. So this is the performance of the model on the 4 different data sets. And this is probably the highest quality data set, Leffingwell. You can see that we reach about 0.9 area under ROC. The perfect classification corresponds to 1. So I stretched out the axis a little bit. The random classification is at 50%, so we're somewhere about 80% of the way to perfectly classifying the odorants. And so you can see the crosses here are just different folds. There are 5 different, it's a 5-fold cross-validation. This is the average performance, and this is the network, the GNN that Alex Velichko showed before. So overall, the idea is that we probably have saturated the quality of the data set, so we're hanging out in our performance at around 0.9. This is just analysis of what the network actually does. So individual points here are the smells, and this is the DeepNose features, which means that these are the analogs of olfactory receptors, right? So these are the same smells. You can see here replicated 3 times. And these are all the smells that have a description pear. Well, these are all the smells that have description garlic, vanilla, and so on. And this first dimension most likely corresponds to the pleasantness that I described before, which is like the principal component number 1 and the most important dimension in olfactory perception. And then the idea here is that you can just take the semantic data set and run isomap embedding on it, and you get a very similar representation. Right, so these are like the pictures that you see here are very similar, but there's no neural network training here. So this is a result of training the neural net. So the overall idea is that what network is doing is just it figures out the semantic embedding, and then it learns how to, using convolutional filters, how to project the structure into that semantic space. So if I wasn't interested in the structure, I could have just discovered the space by running like a low-dimensional embedding. Then we also decided to do some neuroscience on those networks and figure out where are those features located. So by running this lesion algorithm, which we we learned from vision. So where you basically, what you do is you occlude certain pixel, and then you see how the channel, how it affects certain output channel. Like in this case, you can figure out that the face of the dog, this label, Pomeranian, is located here by just occluding that particular pixel and observing how strongly it affects the output for that particular channel. Oh, this is where the wheel is located, because the car wheel label is affected by occluding those pixels, right? So we can play the same game with the molecules by just blocking an atom in the molecule and seeing how strongly it affects the output. And these are some examples. So this is a notorious example of camphorous. You know, there are a bunch of images on the internet you can find of molecules that are completely different structure, and they smell camphorous. Even though for this particular label, the area under receiver operating characteristic is pretty close to 1. It's really it is very robust in predicting this label. I'm just showing all the examples. Like here, for example, for naphthalene, our network failed to predict that it's camphorous. It just thinks that it's metallic, right? But for other examples, it's remarkable how well it actually works in capturing different descriptors. So you can compare the actual and the predicted. They're very close. And you can also compute where camphorous is actually located in the molecule. So this is another interesting example, musk, which also is captured pretty well. So the molecules of diverse architecture, they will be predicted to smell musk. So there are some other examples. This is probably my favorite example, where you see the network, the actual descriptors were fatty, green, waxy, and milky, but it didn't give milk, it gave dairy, but I don't think I can really tell the difference between dairy and milky. So my point is that it's fairly successful in just capturing like individual, if you just want to predict the binary descriptors, and in many cases it makes sense. One of the advantages of our network, especially compared to graph neural networks, which don't contain explicitly the 3D information, is the ability to interact with molecules in 3D. So our network will give different labels for molecules which are mirror symmetric. Like, for example, in this example, this is a pair of enantiomers which are mirror reflected images of each other, and it predicts that those are camphorous molecules. It gives a different descriptor, like it thinks that this molecule is tobacco, for example. And even for the molecules that are not mirror reflected, but just the different 3D conformations of each other, it gives different descriptors. So it also can handle mixtures. I don't have time to describe mixtures, but we can also put mixtures into different orientation channels and deal with mixtures this way. Where is this all going is I think, more into the direction of animal, like going towards the animal models. And like one of the limitations, I think, why we saturate this data set at about like AOROC at 0.9, is that it's really not, the language models are not really fully reflecting the perception. I don't think it's possible. There isn't like intrinsic accuracy or like semantic bottleneck in the way how we can describe how things smell. So the mapping between perception or reality to language is really lossful. So we can't really, I don't think we can reconstruct uniquely the perception from language. So we're moving towards animal models. So these are data collected in Dima Rinberg lab at NYU, which is data on olfactory responses in the mouse olfactory bulb, from the surface of olfactory bulb. And you see here embeddings of olfactory space for two different mice. And as you can see, these are representations of the same molecule. They're connected by lines, and they're aligned, bless you, they're aligned very closely to each other, which just means that perhaps the perception of molecules is not so different in your and my brain. And I guess this is the way to look under the hood, instead of using this the filter, the semantic filter, to look at the original signal and trying to see the representations of molecules. And our hope is that we can scale these data sets to higher number of points, so we can actually approach the possibility of training deep learning models. So as a conclusion, I told you about the DeepNose network, which is a convolutional neural net that replicates some aspects of how olfactory receptors interact with odorants. And it's also predictive about olfactory percepts of humans. And DeepNose can discriminate between shapes that have the shared topology, but they have different three-dimensional conformation. As a thanks, I would like to thank this remarkable group of people, which are called osmonauts. So this is people who are enthusiastic about the sense of smell. These are 8 labs. I should say that this is not my lab. There are 8 labs there. It's a project which is funded by NIH, and we're standing here outside the Grace Auditorium right there. It was like about a year ago that we were together. So this is my lab that actually did the work. Sergei Shuvaev, Kristina Samoilova, Kuaitron, and Cyril Moscart. So they are the ones who are actually responsible for the stuff that I showed you in the talk. Thank you very much for your attention.
Thanks, Alex. Questions for Alex?
Great talk, Alex. So in your model, how do you represent the odor receptors, and how do you model the interaction between odor molecule and the receptors?
How will I model them?
Yes, you mentioned that hidden near there is some interaction between the molecule and the odor receptors, right? Just want to make sure.
Well, in principle, architecturally, the model contains filters, like nonlinear filters, because it's a convolutional neural net that interacts with molecules in 3D. So potentially, there's an architectural possibility to incorporate receptor information. At this point, we don't have receptor information, but my hope is that at some point, like while working with our collaborators, like Dino, for example, present here, we have this project of trying to figure out how individual receptors interact with odors. We can actually inject the loss of corresponding to receptor responses in the middle of our model, and hopefully combine different modalities of different data sets to build a better model. So that's why I'm very enthusiastic about keeping it convolutional, so that we can incorporate data. I mean, it's a long game, and we are somewhere in the 4th inning. So hopefully at some point we'll be able to combine more data sets to crack it.
Oh, thanks. Great talk, beautiful stuff.
Thank you.
I have a question about the nature of the way the encoding emerges in the cortex as you go from olfactory bulb, where you've got glomeruli which are sort of dispersed, and it's not necessarily the case that where they are relative to each other has any bearing on anything, but then the map has to be generated, and there is an actual anatomical kind of topography to it in the cortex. Is that right? There's not. Okay, so that's really interesting then. So how do you go from something that doesn't have an anatomical topography to a perceptual map, which I presume is necessarily contrastive, right?
Yeah.
In the sense that one odor, the position of an odor in the map, also is implied by what it's not. Right? Similar to what.
So there is a
topography.
There's topography in the perceptual space, some degree of topography in the perceptual space for sure, but in the cortex, I think it's still not clear. But overall, it looks like cortex is just a giant recurrent structure. Like in the zeros approximation, it's a giant recurrent structure, and it appears that odors evoke like distributed responses, and the projection from olfactory bulb to the cortex is very diffuse. For example, one glomerulus can project all over the cortex. And I think you are asking a question which is really on the forefront of olfactory research. What happens? Why do you have a precise map in the glomeruli? By the way, the receptors are mapped in very stereotypical fashion from individual to individual, and then you go to this similarly disordered map in the cortex, and then you combine it in the perceptual map, which has a resemblance of topography. So this just blows my mind. I don't know.
The idea of scaling up this problem by getting data from animals is very exciting. I'm interested in learning more about your thoughts on sort of aligning what you're going to learn from the maps that animals use with the humans. It's easy to sort of maybe see how what you do where these do intersect, like the Venn diagram intersection, but what about the parts which are unique to each?
Yeah, I mean, we have a strategy there. Interestingly enough, there is actually work by Rick Gherkin and Joel Mainland, which was published a few years ago, where they show that you can align at least one dimension in perceptual space of mice and humans. Like within this collaboration which I presented, this this remarkable group of people, we find the second dimension. So when humans compare perceptual space and compared to mice, 2 dimensions are conserved. So in principle, you can use mouse data to train at least 2 dimensions in the human olfactory space. But I'm also I'm very optimistic about like learning the logic of receptor to molecule interaction from the mice and somehow transferring it to human receptors. So, which could be done. So there are multiple avenues how we can combine these pieces, both on the level of perception and individual ORs, individual receptors.
Hi, can you hear me? Okay. A lot of the descriptors in the semantic data set are food related, and a lot of the orders also have ethological relevance to nutrition. And there's this other chemosensory system which has a lot of overlap with the olfactory system, the gustatory system, and there is this diffuse the sensing to representation. So, and there is a lot of interaction between the olfactory system and the gustatory system. So I was wondering if you might get a fuller picture if you instead talk about the flavor code as opposed to just the olfactory code.
Yeah, I mean it's an excellent idea. We're thinking about doing that. It definitely needs to be done. There's not much data about like the flavor, like the interaction between olfaction and taste or flavor, like forming a flavor percept. And this is like one of the reasons why olfaction obviously is so important. We figured it out like during COVID, as many people who had became anosmic due to COVID, they also had a kind of bland sense of taste. So there is a very strong interaction between those 2 modalities. I think like taste, this bland word is maybe the key. Taste is a bit more impoverished in terms of percepts than olfaction, and there are 5 labeled lines that are. So I think it's simpler to crack. Like so, taste is more or less, at least my subjective opinion, is more or less similar to say color space, because there color space we have 3 labeled lines, in space we have 5 labeled lines. So olfaction is a bit more tricky because it's a curved manifold and there is no direct mapping between the responses of one receptor to a particular perceptual variable, right? So it may help. I agree with you, it may help, but I think the help will be marginal. Perhaps it will push predictor by 1%. So I'll be very excited in incorporating it if we find any data on it.
Hi, I love the talk.
Thank you.
I was wondering if you could just give an intuition why you have position encoding for the olfaction. Presumably a receptor is only like so wide and there's only so many spaces a molecule can wind up on it. So why do you need those additional 3 parameters to find the position of ascent?
You mean the 4 pixels that I mentioned? So why we have 4 pixels? Well, there's actually 8, right? So we differentiate between nostrils, but also receptors that are expressed in a zonal fashion in the epithelium. So there are four different, at least in the mouse, like in the rodents, there are 4 different, roughly regions where like each receptor would choose one of those regions where it gets expressed. So in principle, we can differentiate spatial location, and it gives us roughly the idea of how high, like the smell is located. Like, I'm not sure if there is like really direct evidence that it's used behaviorally, but anatomically it's present. So that's why I'm pointing out. So if I count the number of retinal ganglion cells in the retina, it will be like like about 1 million, and that's roughly the resolution of the eye. So perhaps if there is this separation or segmentation of olfactory epithelium into 4 zones, it is used somewhere.
Hi, thank you for the talk.
Thank
Very
you.
interesting work, obviously, with smell. I wanted to ask about the learning objective, but first, could I clarify, is the learning objective to match one particular label, like in a vocabulary of labels? Is it like classification? Is that right?
Yeah.
Yeah. I guess I was thinking about this earlier as well. I think there was another person who introduced smell, Osmo AI. And I was wondering, like, because these labels are sort of annotated by these labelers, and I think he even showed that often the labels are noisy, which makes sense. I mean, human raters, like, it's hard to, like, uniquely distinguish, like, this smell is this versus that. It seems sometimes like the answer could be like a blend of smells, or maybe there's not one right answer. And so given this, I'm wondering if you've considered, or the team has considered, also learning the embedding of the label. So, like, in particular in, like, image classification, there has been, like, I think substantial improvement when people have gone beyond trying to match a particular label, but also including sort of like the learning of the embeddings of the labels and, like, doing some kind of, like, for example, clip, and doing some kind of, like, a representation matching. I'm wondering if that's been considered, given the case that, like, maybe it's not just a one-to-one kind of direct matching, or there's a one unique solution.
Yeah, I mean, we've done some, like, limited work in that direction. Like, I showed you high quality data set that includes like 100 labels. That is like a golden standard, right? And people paid really close attention of how to actually choose those words. So there's a research that goes like kind of in quasi-olfactory semantics, which leads up to that atlas of odor character profiles that finds the labels that are really, really descriptive, and the small number of labels, 145 or so, right? But so there is substantial research which went into that data set. So it turns out that the data sets that includes 1,000 smells, but with those like noisy binary labels, gives you a way better predictor than like 100 smells with very, very well-defined labels. So this is an example that like a lot of very imprecise data is better than having like a small number of very precise data, I guess. There is we try to do like language embedding. We try to do just represent molecules, descriptors as word-to-vec representations. But it doesn't give much improvement, unfortunately. So it turns out that just like brute force training for the labels is working almost at the same level as, I think they're just statistically, it's a statistical tie. So we abandoned that direction because it was more complex. And I think the reason why is because the model itself can figure out the embeddings. As I showed you, that space, where it actually does train the embedding, which is relevant to olfactory system, as opposed to using word-to-vec representation, right? Which is like more separate from the olfactory system. So, to be fair, to be clear, we train like all 4 data sets in the same network, right? So the convolutional layers, they learn embedding for all 4 data sets, and we just use different heads for different data sets. So we embed all the labels simultaneously, and I think what it does, it just learns the correct embedding first, and then it learns to go from structure to that embedding. There's also a long history of different methods of measuring smells. I can tell you, like in a coffee break, if you're interested.
One final question over there.
I've got the final question. I had one of the first questions. That was a really beautiful talk,
Thank
and
you.
I was also delighted to hear for the first time mentioning of synesthesia, because I've been wondering over all of the sensory perception and the modeling in AI, or how this would work. The basic assumption underlying from with all speakers, as far as I got it from them, was like that vision is vision and auditory input is auditory input and olfactory input. It's smell, and it has like a neural substrate in the brain. But how about synesthesia, which does exist, and you can make it visible with diffusion tensor imaging? What are the? It's sort of a black box in this whole sensory input AI trajectory, but how could you account for it that this is happening? I have no idea whether this exists in animals, but in any case, it exists clearly in human individuals.
Yeah, I was as much surprised as you. I mean, when this kind of emerged, when I found this book where this perfumer was actually using synesthesia. And as far as I talk to people, there are papers that study this synesthesia, like specifically odor to frequency synesthesia. And apparently this olfactory pitch, let's call it, is not absolute. It's relative. So there are people who just present you a smell and then they ask you to pick, like using some binary search, to pick a corresponding tone that corresponds. And apparently it's basically transferable across people. And it's also, if you can use different starting tones, and the relative arrangement of smells will be conserved, but depending on the starting tone, the absolute frequency will be different. So it's a relative pitch. It's sort of like music, right? So this is like a remarkable phenomenon. It's completely understudied. Also, I find, like, by just talking to people, this is like one of the most dominant synesthesias. So probably 90% of people, if I asked what is lower in pitch, oil or vinegar, will tell that oil is lower in associated auditory pitch. And I don't know how they find it out. So in animal research, there is actually this notion of cross-modal interaction, which occurs in olfactory tubercle. So apparently, olfactory tubercle, it's an area, called, like I think sometimes called olfactory striatum, that contains neurons which are sensitive to both smells and sounds. It's not clear, like what is the significance. I'm not sure what is the behavioral significance of that, but it just remains an interesting curiosity or fact.
Thanks, Alex. Yeah.